Welcome to Creative Biolabs. Creative Biolabs has been a leading bio company in immunotherapy and pharmaceuticals, especially in non-IgG antibody development, through more than a decade of exploration and expansion. Today we will make a brief introduction to non-IgG antibodies and show some related products and services of Creative Biolabs. Part 1. What is non-IgG antibody? Immunoglobulin, Ig, also known as antibody, functions as part of the immune system to identify and neutralize foreign objects such as pathogenic bacteria and viruses. To allow the immune system to recognize millions of different antigens, the antigen binding sites at both tips of the antibody come in an equally wide variety. In contrast, the remainder of the antibody is relatively constant. It only occurs in a few variants, which define the antibody's class or isotype: IgA, IgD, IgE, IgG, or IgM. The five main antibody isotypes are mainly based on the difference of amino acid sequence in the heavy chain constant region of the antibody. IgA is the predominant antibody in mucus secretions such as saliva, tears, milk, and intestinal juice. It is involved in preventing pathogens from entering through mucosal barriers. It can be found in a monomeric or dimeric form. IgD was first identified in patients with multiple myeloma in 1965. It is found predominantly on the surface of B cells, and has a similar structure to IgG, but with an extended hinge region that is very susceptible to proteolytic digestion. IgE has two additional constant domains in place of the hinge region. It plays an important role in defense against parasites, and is also a key mediator of type I hypersensitivity reactions or allergies. IgM is the predominant antibody in the primary immune response, which can present as a pentamer or hexamer in solution and as a monomer on the surface of B cells. Part 2. Introduction to IgA. In human body, the IgA content in serum is the second highest, only lower than that of IgG, accounting for 10 to 20% of the total immunoglobulins, and has a unique role in mediating immunity. IgA is a polyvalent antibody, which is abundant in mucosal secretions. As the first line of defense against infection, IgA is the main defense mechanism for mucosal infections. Secretory IgA functions to prevent foreign substances from entering the circulatory system, rather than to destroy antigens. The monomeric structural unit of IgA comprises two identical heavy chains and two identical light chains arranged into two Fab regions and an Fc region, separated by a flexible hinge region. The heavy and light chains fold up into globular domains. IgA mainly exists in the form of dimers in secretions, and S-IgA is formed by the polymerization of two or more IgA monomers. A typical S-IgA is mainly composed of two IgA monomers, a J chain, and a secretory component. The secretory component is a 75 kilodalton polypeptide chain synthesized by intestinal epithelial cells, which acts as a dimer IgA-specific receptor for binding to the dimer form of IgA. The total molecular weight of IgA antibodies is approximately 160 kilodalton. There are two isotypes of IgA, IgA1 and IgA2, both of which are highly glycosylated proteins. The proportion of IgA1 and IgA2 secretory cells in different lymphoid tissues varies from each other. IgA1 is dominant in serum, accounting for 85% of the total IgA concentration in serum. IgA1 shows broad resistance to several proteases, but some can affect or splice on the hinge region. IgA1 shows a good immune response to protein antigens, but a low degree of immune response to polysaccharides and lipopolysaccharides. IgA2 accounts for only 15% of the total IgA in serum, and it mainly exists in the mucous membranes of the airways, eyes, and gastrointestinal tract to combat polysaccharides and lipopolysaccharide antigens. It also shows excellent resistance to proteolysis and many bacterial proteases, and plays an important role in fighting bacterial infections. The intestine is the largest lymphoid tissue in the body. One striking feature of intestinal immunity is its ability to generate great amounts of non-inflammatory IgA antibodies that serve as the first line of protection against pathogens. The basic pattern of IgA production involves the stimulation of mucosal B cells in the Peyer's patches, the circulation of IgA-committed plasma cells via the bloodstream and homing to the intestinal mucosa, and local antibody synthesis for export through the intestinal membranes. Multiple cytokines, including TGF-beta, IL-10, IL-4, IL-5, and IL-6, are required to promote IgA class switching and terminal differentiation process of the B cells. Secreted IgA promotes immune exclusion by entrapping dietary antigens and microorganisms in the mucus and functions to neutralize toxins and pathogenic microbes. Part 3: Introduction to IgD. IgD is an antibody synthesized late in the ontogeny. The level of IgD is low in serum, which is 3 to 40 g per milliliter, and it accounts for about 0.25% of the total serum immunoglobulin. It is composed of 2 light chains and 2 heavy chains. The heavy chain and the light chain are connected by disulfide bonds, forming a conformation similar to the shape of the letter Y. The hinge region of IgD is long and is sensitive to protease hydrolysis, resulting in a short half-life of only 2.8 days. There are 2 types of IgD: serotype IgD, also known as S-IgD, and membrane-bound IgD, known as M-IgD. It is now believed that S-IgD can recognize soluble antigens to promote a protective humoral response, and M-IgD constitutes B-cell receptor, which is a sign of B-cell development, differentiation, and maturity. Secreted IgD exists and plays an elusive function in blood, mucosal secretions, and on the surface of innate immune effector cells. IgD apparently is also able to bind to basophils and mast cells and activate these cells to produce antimicrobial factors that are functional in respiratory immune defense in humans. Transitional B cells emerging from the bone marrow differentiate into mature follicular B cells that colonize the human aerodigestive mucosa, the mouse nasal-associated lymphoid tissue, and the mouse mesenteric lymph node. At these sites, some follicular B cells enter a germinal center program that fosters class switching from IgM to IgD. The resulting IgD class switch B cells differentiate into plasma cells that release IgD both locally and in the circulation. By recognizing non-invasive commensals, invasive pathogens, and probably airborne and food antigens, secreted IgD may enhance immune homeostasis, tolerance, and surveillance at mucosal sites of antigen entry. Engagement of basophil-bound IgD by antigen would then trigger basophil release of IL-4, which may amplify humoral immunity by enhancing B-cell production of IgG. This antibody is abundantly produced in human nasopharyngeal cavities and may cooperate with IgD to provide local immune protection. Part 4: Introduction to IgE. IgE is an antibody that has been found only in mammals. IgE primarily defends against parasitic invasion and is responsible for allergic reactions. Monomers of IgE consist of 2 heavy chains, epsilon chains, and 2 light chains, with the epsilon chain containing 4 Ig-like constant domains, from C epsilon 1 to C epsilon 4. IgE is considered to be an important component of the immune response against certain parasitic infections, including Schistosoma mansani, Trichinella spiralis, and Fasciola hepatica. IgE is also used in the immune defense against certain protozoan parasites such as Plasmodium falciparum. The pathogenesis of IgE-mediated food allergy is divided into 2 phases, a sensitization phase and an elicitation phase. The allergen is taken up by antigen-presenting cells during the sensitization phase, resulting in the activation of Th2 cells, which then contribute to the activation of B cells, and proliferate into IgE-secreting plasma cells. This forms the basis for the cell-free IgE-based in vitro test methods as well as the IgE epitope mapping-based tests, with the IgE from the food allergic patients. In the elicitation phase, allergens may, upon re-exposure, cross-link FCE or I-bound IgE on mast cells and basophils, leading to mediator release and symptoms similar to food allergy. Part 5: Introduction to IgM. IgM is the largest antibody that is produced by the immune system of vertebrates. It can attack against a wide variety of antigens and take part in many steps of the primary and adaptive humoral immune response. The predominant form of IgM in human serum is pentamer, which is composed of 5 monomers connected with a joining chain. The other forms, such as hexamers and monomers, can also be found at rather a low abundance in vivo. IgM can play diverse roles in health and disease. Whereas extensive studies have documented the contribution of IgM as the first line of defense from infection, more recent reports have highlighted their potential roles in the maintenance of immune homeostasis, prevention from overexuberant inflammatory responses, and development of autoimmune disease. The role of IgM in tissue homeostasis involves the activation of complement pathways and aids in the prevention of inflammation, autoimmune, and cancer. These functions include recognizing apoptotic cell membranes and supporting phagocytic cell clearance of apoptotic cells, clearing changed or malignant cells or misfolded proteins, and regulating disease-associated IgG and B cell clones. This figure summarizes the differences in properties among the 5 immunoglobulins. Part 6, Related Services and Products. As a senior service provider, Creative Biolabs has accumulated years' experience and acquired reputations in antibody development. Possessing experience and well-established platforms, we have successfully constructed a novel service system to provide comprehensive non-IgG therapeutic antibody development services, which include but are not limited to non-IgG therapeutic antibody discovery. To discover more efficient therapeutic antibodies and provide more comprehensive services, Creative Biolabs has developed the exclusive non-IgG therapeutic antibody platform based on our acquired experience and accumulated expertise. After years of efforts and concentrated study, we are confident in providing a full range of custom non-IgG therapeutic antibody discovery services for international clients with the best strategies and professional solutions. Non-IgG therapeutic antibody engineering. At Creative Biolabs, not only IgG antibodies, but also non-IgG antibodies are well developed. Our professional scientists optimize non-IgG antibodies to obtain better efficacy by innovative engineering technologies, such as humanization, glycosylation, and conjugation. Non-IgG therapeutic antibody production and purification. Due to their different characteristics, the production and purification methods of non-IgG antibodies are different and even more difficult than those conventional antibody production and purification technologies. These difficulties are also some of the critical factors hindering the development of non-IgG antibodies. It is exciting that Creative Biolabs is the one who is capable to support our clients in non-IgG antibodies production and purification using our abundant experience and expertise in this field. Non-IgG therapeutic antibody characterization. Antibody characterization is an important process to generate an antibody with dependable, predictable, and intended performance. Creative Biolabs has developed several technologies to evaluate the quality, quantity, and function of the produced non-IgG therapeutic antibodies, such as ELISA, the abbreviation for enzyme-linked immunosorbent assay, flow cytometry, immunoblotting, SDS-PAGE, the abbreviation for sodium dodecyl sulfate polyacrylamide gel electrophoresis, high-performance liquid chromatography, and mass spectrometry. Non-IgG therapeutic antibody PARC/PD evaluation. Creative Biolabs conducts PARC/PD assays to analyze and evaluate therapeutic non-IgG antibodies immediately after they are produced, purified, and characterized by our comprehensive technique platform. Integrally understanding PARC/PD characteristics of non-IgG therapeutic antibodies, including exposure of action site, target occupancy, and performance of pharmacological activity, is of great significance in achieving the success for further applications. In addition, we have invested a great deal of talent and effort to successfully build a comprehensive technology platform, aiming to provide the most diverse non-IgG antibody products to global customers. Here, we offer a wide range of high-quality off-the-shelf non-IgG antibody products, mainly IgA, IgM and IgE monoclonal/polyclonal antibodies, as well as a variety of custom antibodies and assay kits for academic and commercial use. For more details about Creative Biolabs, please visit our website. Thanks for listening.